Hello everyone and welcome back to Creation Myths. Today I have a video that I'm kind of disappointed I had to make it. I was reluctant to reach the conclusion that I have finally reached. So today I have to say, okay, I was wrong. I think Dr. Casey Luskin from Discovery Institute is just lying. And again, I was reluctant, I was hesitant to come to this conclusion. A lot of other people came to this conclusion before me, and I have to say to all of you, you were right. So let's talk about it. On July 19th, 2024, I released this video in response to a podcast interview done by Discovery Institute's Dr. Casey Luskin. I called this video, Dr. Casey Luskin Misunderstood My Whole Deal, because in that interview, the host asked him specifically about some things that I said during our May 2024 debate on junk DNA. And Dr. Luskin did not seem to understand the arguments I was making. He presented my arguments as, in some cases, the direct opposite of what they actually were. Now, I made clear in this response video, linked down below, that I think Dr. Luskin was making very bad arguments, but he was making them honestly. He was not intentionally trying to misrepresent the arguments I was making. He did not understand what I was trying to convey and was responding to something else due to that misunderstanding. And I even offered a couple of potential explanations for, like, maybe you thought because we were talking about this thing, you thought it applied to this other thing, and that's where the confusion was. So I think he was making bad arguments honestly in that video. That is what I portrayed in that response, which again is linked down below if you want to catch up on the backstory here. Now, a lot of viewers who left comments disagreed with that assessment. A lot of people basically said, look, it's really good that you're giving him the benefit of the doubt, but I, the people who were commenting, think he's being dishonest. He's deliberately misrepresenting your arguments. And when I say many viewers disagreed with my take on this, I mean many people left comments disagreeing with my, as people said, generous interpretation of Dr. Luskin's interview. Many viewers. So now we have this article published on the Discovery Institute's Evolution News blog on August 5th, 2024, authored by Dr. Casey Luskin, called Disease-Associated Junk DNA is Evidence of Function. And it's this article that makes me now reluctantly agree Dr. Luskin is just being deliberately dishonest. Like, at this point, I think he's just lying. So we're going to go through why that's the case, because I don't make this judgment lightly. I really do try my best as, not just with Dr. Luskin, but with anybody that I'm interacting with or critical of. I really do try to start from a place of assuming good intentions and good faith in whatever argument or debate that we're having. So it takes a lot to get me to this place where I'm willing to conclude that somebody is being deliberately dishonest, but I think this article puts it over the line for me with Dr. Luskin. So let me show you what's going on here and explain how I got to this place. In this article, Dr. Luskin repeatedly conflates non-coding, which we mean non-protein coding DNA, with junk DNA, DNA that does not have a function. So here are some examples from that article. How do we determine when a segment of non-coding junk DNA has function? One of the most common functions of non-coding / junk DNA is to regulate gene expression. Gene desert, i.e. non-coding or junk DNA region. We're conflating these two things quite directly here. We're saying that non-coding, by which he means non-protein coding DNA, is the equivalent of junk DNA. The idea that he's trying to get across here is that biologists at one point characterized all non-protein coding DNA as junk DNA. That is not true. I'll talk more about that in a couple of minutes. But that's what he's trying to convey here. Now, I've covered this point specifically. In December of 2023, I put out This video, Creation Myth, biologists thought all non-coding DNA was junk. And in that video, I actually cited two people from Discovery Institute, Dr. Jonathan Wells and Dr. Douglas Axe. Now, my suspicion is that Dr. Luskin is getting this claim from them or potentially other colleagues at Discovery Institute. Okay, that's where I think he's getting this, which is why, up till this point, I was willing to kind of let him slide on this, because it's not his field. He's not a biologist. He's not a geneticist. So if the actual biologists at Discovery Institute are telling him this thing, this incorrect thing about the history of junk DNA, then he's going to take that at face value. That's what I would expect. So I was willing to say, okay, he doesn't know, it's an honest mistake, okay? But here's the thing. At this point now fast forward from, December 2023, earlier in 2024, now we're in August of 2024, I don't think that I can extend that charity any further. In the lead up to and also since our debate, we've, corresponded via email. And when that podcast interview went up, as I record this a few weeks back, I reached out to him to express my surprise that he did not accurately represent and seemed to be misunderstanding some of the arguments I was making. Now, in that email to him, I added this little bit at the end. I said, there was one more thing, a very small thing, but I do want to mention this. Right at the start, you said something to the effect that for decades, we, biologists, thought 98% plus, that was the phrase you used, of the genome was junk, referring to the non-protein coding fraction. That is a frequent claim for a lot of people, including your Discovery Institute colleagues, and it is simply not true, has never been true, and the reference provided in one of the Evolution News posts in this back and forth in support of that claim just references a recent paper that also mischaracterized the history. The reality is that the term junk DNA was first defined in the literature in 1972. But by then, we had documented a whole lot of functional non-protein coding DNA. For example, tRNAs, ribosomal RNA, promoters, centromeres, and telomeres. So it has never been the case that the term referred to all non-protein coding DNA. I sent this email to Dr. Luskin on July 19th, 2024. But now, August 5th, 2024, we have the article that I showed you a few minutes ago where he repeatedly conflates non-protein coding DNA with junk DNA. The conclusion that I'm forced to reach here is that Dr. Luskin is lying about the meaning and the history of the term junk DNA. That's what's going on here. I can see no other explanation for what's written in this August 5th article on Evolution News. So why is Dr. Luskin doing this? What's the point here? It's actually a really simple argument that he's structuring, and here it is. Step 1, all non-protein coding DNA was originally classified as junk. Step 2, every function found in non-protein coding DNA is a function for junk DNA. Step 3, therefore, we should conclude that virtually all DNA in the human genome has a function. That's the argument. That's what this bait and switch accomplishes. And I want to be very clear. This changing of the definition and the history of the term junk DNA to mean all non-protein coding DNA, this is a deliberate choice. This is a bait and switch because it allows Dr. Luskin to set up this argument like this. Without this, you can't point to all of these different functional bits of the genome that are non-protein coding and use them as evidence for the argument that there is little or no junk DNA. By changing the history of the term junk DNA, it allows you to structure the argument like this. That's what's going on here. And I want to make sure that's clear for everybody. This is not an arbitrary bit of misinformation. This is not lying for no reason. There is a purpose here, and it is to structure this specific argument. Now, again, I want to make perfectly clear. This is fundamentally dishonest. I cannot imagine that Dr. Luskin does not know better at this point. It would be astonishing to me if he genuinely thought this was an honest thing to say. So the only thing I can conclude is that he's lying about it. That's where I land, and I don't see any other choice, as much as I didn't want to land here. Here I am. That's just how it is. Hey, but since we're here, let's address his arguments anyway, as though they're being made in good faith. I'm going to show you a few examples of the types of arguments he makes in this article and explain why he's still wrong. We're going to look at his examples of functional junk DNA, and we're going to see why the arguments don't work. First, he uses enhancers as an example. Enhancers are regions of the genome where transcription factors bind. These are regulatory proteins. They bind to the enhancers, which are regions of the DNA, and when they do so, they upregulate the expression of nearby genes in the genome. So he's pointing out that you have certain non-coding regions in the genome, like this region he calls out here, chromosome 21q22. It's a gene enhancer that is sometimes deleteriously affected when mutations arise in this junk region. He talks about another example that regulates the gene ETS2, but here's the thing. He keeps saying junk DNA elements contain enhancers. This enhancer that's in this junk region. These words only make sense in the context of the bait and switch I just described. In real biology, a junk DNA element cannot be nor contain an enhancer because enhancers are functional, and that exists independent of the context of where they're found. So you could have an enhancer that's like part of a line element, which is a type of retrotransposon, or you could have an enhancer derived from an endogenous retrovirus. If a region is functioning as an enhancer, we're going to say, aha, that's functional, no matter what it's derived from. So these enhancer regions, which make up a very small fraction of the genome, less than 1%, those are functional. And it literally doesn't make sense to talk about a junk DNA that contains an enhancer or an enhancer junk DNA region, right? Those things don't compute. They don't go together, because once we identify regions as enhancers, which we can do very readily based on transcription factor binding and selection to maintain that sequence, right? We can identify the enhancers that are being used. We can put those in the functional bin. Doesn't matter what else is going on or where they come from. Those are functional. So that's not a very good example of a junk region that has some surprising new function. He also uses the example of genes derived from endogenous retroviruses. So HERVs are human endogenous retroviruses, and he says HERVs from the families W and FRD encoding ENV, that's a gene, ENV, play a fundamental role in cellular fusion during the formation of the placenta and are now annotated as the syncytin 1 and syncytin 2 genes. Here's the thing. Syncytins are protein-coding genes acquired from retroviruses via a process called horizontal gene transfer. In this particular example, virus integrated into the human genome, and then we started using one of those viral genes for our own means, right? This cellular fusion thing that goes on during placental formation is not something the virus does. That's us co-opting a viral gene for a new function. So this is not an example of a functional ERV. This is an example of acquiring genes from retroviruses via horizontal gene transfer. Importantly, syncytins were never considered junk because they're protein-coding genes, which is like the simplest 30,000-foot test for whether or not a region is functional. Does it get translated to a polypeptide? Then pretty decent chance that thing is functional, and most people are not going to object if you tentatively put it in the functional bin. Nobody's going to complain about that. I don't think anybody's ever called syncytin 1 or syncytin 2 junk DNA. That would be silly because, again, they're protein-coding genes. And you know who agrees with me about this? Dr. Casey Luskin, because this is his standard. This is the thing he does wrong up above. He repeatedly conflates non-protein coding DNA with junk DNA. Even under his incorrect definition of junk, where all non-protein coding DNA is junk, these genes would not be considered junk. So Why he's using them as an example of functional junk DNA, I don't understand. I mean, I do. It's because they're derived from endogenous retroviruses, so he thinks he can use them as an example of functional ERVs. But that doesn't make sense because they're not like intact ERVs sitting in our genome. They're genes that we've acquired that we regulate using our non-ERV regulatory things. And I think part of what's going on here, again, is Dr. Luskin is not a biologist, he's a geologist, so he doesn't know the intricacies of this stuff. So maybe you can get away with calling this what ignorance rather than dishonesty, but like, I think that's pushing it. He also points out that endogenous retroviruses are associated with psychiatric disorders. So this top quote here is from Dr. Luskin's article. He says, it looks like these ERVs have some kind of function in the brain, but when their normal expression is disrupted, psychiatric diseases result. And the paper that he cites for this contains these little bits that I'm quoting right here. We identify 1,238 human endogenous retroviruses with expression regulated in cis, of which 26 represent expression signals associated with psychiatric disorders. Further down, they wrote, in total, we identified 26 HERV expression signatures associated with psychiatric disorder susceptibility. In other words, these endogenous retroviral sequences were being expressed at abnormal levels, and that was associated with psychiatric disorders. This shows the opposite of what Dr. Luskin claims. He says this shows that those ERVs are functional. What this actually shows is that they're not, because if they're normally suppressed, and then they're expressed, right? They break out of that suppression that we're imposing on them, and that causes psychiatric disorders. That means they're not functional. That means they're not doing a job that helps us. They're hurting us when they're allowed to be biochemically active. That's the opposite of function. Now, this counter-argument, the idea that they're associated with disease indicates that they're non-functional, is not new to Dr. Luskin. He says, well, I have heard it argued that finding that junk DNA is related to the cause of some disease is not evidence that it is functional. I have heard it argued. Where has he heard that? He's heard it from me in our debate on junk DNA from May of 2024. That's where he's heard this before. Watch this clip. I directly brought this up to him. Check it out. I do just want to say on the disease thing, I think that's extraordinarily strong evidence that these sequences are non-functional, because a lot of what happens with these, it's going to be things like it's retrotransposons, it's going to be SVA transposons. These are active transposable elements in the genome, and our genomes work really hard to shut them down. So they're not doing their jumping gene activity. And when they mutate, they evade our kind of countermeasures, and they mobilize. And that's been shown to be associated with cancers, with neurological disorders, developmental disorders. And so it's not the case that that indicates that they're functional. It shows that they're actually harmful to you when they are biochemically active, and we do a lot of work in our cells to make sure they're not. So what am I supposed to make of this? Because it seems like he makes a claim, right, that they're functional because they are associated with disease when they get out of regulation. And I explain to his face, no, that shows that they're non-functional because they're being suppressed, and when they lose the suppression, that leads to disease. Does he respond to that at all? No. Does he seem interested in responding to that at all? No. That does not strike me as an honest approach to this conversation. And possibly now I'm being too harsh, but I feel like maybe I've been primed to see things in that light, given what we've already talked about so far in this video. The last example I'll show you is, I think, the most egregious. In this example, Dr. Luskin is talking about viruses integrating into the human genome and disease, specifically cancer, resulting. So specifically here, he's talking about the human papillomavirus, the main cause of the disease, cervical cancer, embeds itself in this particular part of the genome. In doing so, the virus enhances a gene called MYC, or MYC, which is a well-known driver of cancer. Studies are suggesting that the connection between this region of the genome and the gene may also play a role in a number of ovarian, breast, prostate, and colorectal cancers. So what's going on here is a virus inserts in the human genome, acts as an enhancer, causes cancer. Therefore, the insertion site, which before the virus inserted lacked an enhancer, is functional? We're talking about a viral sequence insertion occurring within an individual's lifetime. An individual gets infected with human papillomavirus or Epstein-Barr virus. There are a bunch of viruses that do this. And those viruses insert in regions where they drop enhancers that upregulates a nearby gene, and then the upregulation of that gene is a risk factor for certain cancers. What does that have to do with the target site in the human genome? It's not like there's something there that's getting broken, right? We're talking about the virus enhancing a nearby gene. So the virus, by inserting in that region, is upregulating the gene. It's not the human genome that's doing it, right? It's not a pre-existing part of the human genome that's causing these cancers. It's a virus inserting and breaking the regulation of a gene by causing it to be upregulated with a viral enhancer. How is this possibly evidence for function in that insertion region? That makes no sense to me. I cannot wrap my head around this particular example. Maybe somebody in the comments can explain to me what the logic is supposed to be here. Because really, like speaking directly to Dr. Luskin, I am not going to insult your intelligence by portraying what we just saw with the viral insertion thing as a real argument that you are making for functional junk DNA. I have too much respect for you to think that that argument is something you actually believe supports the notion that junk DNA regions or non-coding regions, whatever you want to call them, are actually functional. Taking a region that is devoid of things like enhancers, dropping a viral enhancer in there, I refuse to believe that you, Dr. Luskin, think that that indicates the underlying target region is functional in the absence of the viral insertion. That my brain cannot do that trick. I refuse to believe that that is what you think, which means I am forced to believe you are misrepresenting these things to make your point. That is only slightly more attractive as an explanation. I don't want to think that, and in our months-long back and forth here to date, I had not thought that. But that is what I am obligated to believe is going on here. I cannot make my brain believe that this is a real argument you are making. I can't do it. In conclusion, at this point, I don't see an alternative to the belief that Dr. Luskin is lying about non-protein coding DNA and junk DNA and what those words mean and their historical relationship with each other. And also the belief that he's presenting arguments that he knows are not valid. I don't see an alternative to that either. I arrived at this position very reluctantly, and it took me longer to get here than a lot of people watching our months-long interaction have taken to reach that same conclusion. But I think at this point I'm justified in this conclusion, and that's very disappointing to me because really up until just this week, my position was that Dr. Luskin was making bad arguments, incorrect arguments, but he was doing so honestly and in good faith. Based on this most recent article, I no longer believe that is the case. I think he is being deliberately dishonest here. And again, that's disappointing to me because I don't like to believe that about people, but I don't know what choice I have at this particular point. So that's where I'm going to leave it for today. Thank you all for watching. Please, before you go, hit the like button, leave a comment, subscribe, share this video. If you are so inclined, if you appreciate what I'm doing here and are feeling generous, please become a channel member. Members get immediate access to pre-recorded videos instead of having to wait for the public release. Thank you all again for watching, and as always, don't get fooled.